Hello, this is Miss D, and we are going to be talking about how things can go wrong. And so we are talking about mutations, and mutations ultimately are going to be changing the genotype, the DNA. But what we want to talk about is how this can affect the amount of the protein or gene product and what the effect then causes to the phenotype. So we're going to look at the types of mutations and then what the types of effects that can happen. So let's go ahead and get started. We have a picture on the right where I have used some circles, and what they refer to is back to this original DNA strand. Now, mutations alone are simply changes in the DNA sequence. Something is happening to change the order of the base pairing. And you're going to see that has a variety of effects. So, mutations can be classified based on the type of change, and then they can be classified based on the effect that they have. And so let's take a look at that. So point mutations are where we only have one base affected. And so naturally we see it as a point in the DNA. And so these can also be called substitutions. And usually what we're talking about is substituting just one base pair or one nucleotide. And so let's take a look at these. So here is a wild type DNA and the mRNA that's transcribed from it. And so you can see, let me go ahead and get my pointer going. You can see that if we take a look at that second codon here, we went from AAC, and if you notice in the part B, it changes to AAT. Now, if we look at the mRNA, UUG gets changed to then UUA. Now, if you look at the codon chart, the big takeaway here is it did not change the amino acid. So we would call this a silent mutation. The DNA, the RNA could be changed, but the amino acid, and this often happens if it's that third base in a codon, because remember there's some redundancy in the mRNA codon chart. There can be more than one codon, and often it's this third base, that third nucleotide that has that wobble. It can change a little bit. So a silent mutation simply means while we have a difference in the DNA, the amino acid doesn't change. Keep this in mind when we take a look at differences in the genetic differences in animals and plants. They might have slightly different DNA, but their proteins might be the same, and that's because of the silent mutations. We could have a missense. Now, a missense, notice we go from AAC in our wild type to AAG. Now, that's going to change our mRNA to UUC, and that does change the amino acid. And so that changes one amino acid, and so that is called a missense. It doesn't change the rest of the mRNA strand, and therefore it doesn't change the rest of the amino acids. And I think of this as just, okay, well, we made a mistake. And so you think of missense, mistake. There's just one mistake. Now, depending on the change of this amino acid, is if this changed it from a nonpolar to a polar, or a polar to a completely charged amino acid, positive or negatively charged, maybe it went from a positive negative charge to a nonpolar, this could have a drastic change, and we'll take a look at an example of that. But it may not be a big change, and depending on the placement of this amino acid in the protein, it may not be that big of a deal, right? It may not, if it's in an active site of an enzyme and it changed the shape of the active site, then yes, that could be a big deal. But if it's on the outlying structure of the enzyme, it may not be that drastic a change of the shape. And then it could be a nonsense. And so it changes to a stop codon. And so you'll notice we went from uh, um, uh, UGG to UGA, and that's a stop codon. And that's called a nonsense mutation. And and depending on where that occurs, it could stop the amino acid chain very early, in which case you've made a nonsense protein. That's how I like to, think about it. Okay, well, I've just made nonsense. Think about a section in Shakespeare where the soliloquy just stops, and you'd be like, well, that didn't make any sense. Well, it's nonsense. Now, a frame shift is where we insert or delete nucleotide or one or two nucleotides. And if that happens, that's going to be very, very disastrous because let's take a look down here. If we insert a nucleotide, remember how the ribosome reads the mRNA is through the codons. Every three is read and interpreted as an amino acid. Now, if you add one, that changes what we call a reading frame. And there's where the frame shift comes from. We've changed our reading frame. And so all the ribosome is going to do is read the new codons. And because that shifts everything over, every subsequent amino acid or codon after is going to change to a different amino acid. And so a frame shift is, again, an insertion or a deletion of a base. It could be silent, in which case, if it's at the end of the codon stretch, it may not make a big difference. It's going to be a missense for anything after that point, that mutation. And then it could lead to a nonsense as well, because if you shift, you may go from UAC, but then you insert a UGA, and that's a stop codon. So that could lead to a nonsense codon as well. Now, here's a good example. For instance, if we just had this is the normal sentence. The fat cat ate the rat. But if you substitute an H for the F, and notice these are all three-letter words. So this is a nice example. The hat cat ate the rat. And the sentence is mostly making sense except for that word. But notice if we delete one, it shifts everything over, and now we've lost the meaning of the sentence. This would be akin to changing every single codon after that. And same thing if we insert a nucleotide, that's going to be a problem. So we're going to take a look at some examples, and what we want to just take away would be if you change the DNA, that will change the mRNA or tRNA or rRNA. It's going to change the RNA made. Now, changing the mRNA could change one or more amino acid. Remember, it could be silent, it could be a missense, or it could be a frame shift. So that is going to change. Now, if you only change one amino acid, it may not be anything. It could be silent. But changing one amino acid could make a big difference if it changes that charge on the amino acid. So changing one amino acid to another may change positive to negative or negative to neutral. And if that was a significant place on the protein that was a key indication of how that protein folded, that would change how the protein folds, and that's going to affect the function of the protein. So, real important chain of events there. Just follow the logic there. If you change the DNA, it's going to change the RNA. Changing the RNA could change one or more amino acids. That might change the charges involved in folding the protein. and then that could change how the protein then functions. Very, very important, and we're going to take a look at that. Chromosomes can also engage in, can also have mutations, and so you could delete parts of chromosomes. You can actually duplicate part of a chromosome or all of a chromosome, and you can actually add chains to it. This happens during replication events, and you could also then flip flop pieces of the DNA when they invert. And then that we could also translocate, and we already saw that lecture on the translocation for the BCR-ABL protein. So all of these can influence the gene products, and you could make more of a gene, you could make less of a gene product, or it could significantly impair, like the BCR-ABL, that was a translocation, and that made a different protein that caused cancer. So very, very important to understand how these things can cause problems. Okay, so hopefully that gave us a real nice way to start the week.